Assalamualaikum and a very good morning to my respected lecturers. My name is Nur Shazana binti Najmuddin with metric number F17A0092. Today, I'm going to present about my thesis under supervision Dr. Yusrina Andu entitled Morphometric Characteristic and Language Relationship of Mud Crab Scylla olivacea in Kota Baru, Kelantan. We go to the first part, which is research background. Mud crab genus Scylla consists of 4 types of species. The one that I choose to be the sample for my study is Scylla olivacea species. Scylla olivacea, or can be called as orange mud crab, has the smallest size compared to the other species. The color of this species was mixed with red to brown color on its carapace. This species mostly found at the mangrove forests, such as at the area supralittoral and intertidal zones. This because their environment surroundings was appropriate for the mud crabs. It also provides comfortable shelter, can maintain the number of mud crabs, and reducing the percent mortality of mud crabs. So, this is the 24 characters from mud crab body parts. These mud crabs are known as seafood delicacy in Southeast Asia due to its easy availability, has high nutritious value, high demands for food material purpose, whether in local market or in international market. It's because of having good taste and good quality of meat. The first problem from this study was in Kelantan. The study of morphometric characteristic on mud crab Scylla olivacea was very limited compared to the other countries like Terengganu and Sarawak. The second problem was this reduction on size population of mud crabs due to the factors of overfishing give bad effect and may generate continuously catch by fishermen in mud crabs resources. The demand from the buyers that want to use and commercial the mud crabs was high. This is because there were lack of knowledge about the biological development phases. If they gaining and learning the information from the resources conservation, this can help them to achieve a better management in fisheries, and at the same time, this can help support the mud crab populations against the overfishing issues. There are 3 objectives of this study. The first one is to identify the population of mud crab Scylla olivacea at the area of Kota Baru, Kelantan. To determine the relationship between the length and weight of mud crab Scylla olivacea based on its morphometric characteristic. The last one is to analyze the relationship between the morphometric characteristic and the length weight of mud crab Scylla olivacea by using the statistical analysis. Hypothesis of this study: for null hypothesis HO, there's no difference between the morphometric characteristic and length weight relationship of mud crab Scylla olivacea, while the alternate hypothesis HA, there's difference between the morphometric characteristic and length weight relationship of mud crab Scylla olivacea. Next part is literature review. As you can see, there are 4 types of pictures of genus Scylla species, which is Scylla serrata, Scylla paramosi, Scylla olivacea, and Scylla transcubarica. And the right side mentioned that the taxonomy and classification of genus Scylla. Authors found that the population of mud crabs can be affected by the conservation of natural mangrove mud crabs. To differentiate the maturity of mud crabs between male and females, it must look at the shape of its abdomen. The picture on the left side, the abdomen of matured male has narrow and straight, while for the right side, for the matured female, it has globular abdomen which is wider than male. The decline of mud crab population occurs due to the factors of overexploitation and destruction, which can cause the pollution to the habitat surrounding. Next, mud crabs can eat variety of food to improve their reproductive performance, such as maturity, growth rate, gene, and size. Besides, water salinity can give good effect to the mud crab reproduction, like mating process and ovarian maturation. And the standard water salinity treatment that are suited for mud crabs was around 20 ppt to 25 ppt. For the methodology, in this research, there are 30 samples of mud crabs were collected at Pantai Sabak, Pengkalan Cepa, Kota Baru, Kelantan. From the 30 samples of mud crabs, there were 18 of males and 12 of females which come out from various of sizes. This research was conducted starting from 5th September to 14th September of 2020. So, the morphological characters and the length weight measurement of Micropsylla olivacea were analyzed and recorded. To perform this statistical analysis, I have used Microsoft Office Excel because it can summarize all the data obtained. And the statistical analysis actually consists of three types, which is descriptive, linear regression, and t-test. For descriptive statistics, it is used to analyze and represent the data that have been collected. For linear regression, it is to determine the allometric growth, and for t-test, it is used to determine the hypothesis whether it is accepted or not. The pictures above shows that the step on sampling Micropsylla olivacea. The first picture, I will tie the microbes by using raffia ropes to prevent getting injured and for my safety. Second picture is the microbe was weighing by using electronic balance. And the third picture is the microbe was measured by using a ruler. And this is the detailed steps of my workflow study. To the most important part, which is result and discussion. As you can see from the table 1 above, it's shown that the average measurement for both length and body weight of male are higher than female. For example, the average length and weight of male are 60 mm and 182.4 gram respectively. For female, the average of carapace length are 59.5 mm and the average of body weight are 135.75 grams. So here, male microbes are heavier and bigger compared to female microbes. Second table, it is about the equation and correlation coefficient R-squared values between different variables in male of Psylla olivacea. Based on the data in the equation of allometric growth, all these different variables in males have negative allometry since the p-value is less than 1. For R-squared value, it shows that the most strongest relationship goes to the variables between propolis length and propolis width with 0.897 since R-squared value is nearest to 1. And as a result, this is the one of the graph that I randomly choose, which is the relationship between carapace width and propolis length. This table of female Psylla olivacea, which has shows that the relationship was also contains existence of negative allometry. For R-squared value, it is approximately high in all set of the microbes characters relationship, which means that all the relationship of two variables are strong. And there's another one from many graphs which has shown the relationship between carapace width and abdomen width. The equation relative between carapace length and body weight analysis are already illustrated in the table 4. Male and female microbes are positive allometry because the value of B is 6.096 as it is higher than 1. While for R-squared value for these two variables, it is relatively less strong with value 0.59. This is the graph for the relationship among carapace length and total weight for both male and female Psylla olivacea. As you can see in the table 5, the t-value for both male and female microbes are significant at 1% with 2.11 and 2.201 respectively. So, the hypothesis of this study is accepted. To be concluded that this study can facilitate other crab biologists to acquire the microbes' length estimation with exact measurement. Second, it can be utilization resources toward other fishermen in Kota Baru area in the future. Besides, the peoples or readers out there can manage to differentiate the stocks from different species and different geographical. So, this study is very important to the peoples or to help future study in managing the crabs population effectively. That's all from me. Thank you.